Discuss about the histology of the cerebellum. Okay, so this is section through the cerebellum. On the two sides, two hemispheres and midline vermis. Okay, and these are the foliums, and in between the foliums, these are the fissures. Okay, so cerebellum is a highly folded to accommodate millions of neurons in a small area, and folding is called arborvitae, okay, tree like. And cerebellum has two cerebellar hemispheres that are connected to each other by a median vermis, and it is divided into a number of lobules by the fissures. Okay, so these are the lobules divided by these fissures. These depressions are the fissures. Okay, so next, structure of the cerebellum is uniform throughout, that is homotypical. It has outer cortex. Outer cortex is formed by gray matter and inner white matter. Okay, and four nuclei are there. Embedded within this white matter are the nucleus dentatus, globosus, nucleus emboliformis, and nucleus fastigius. Dentatus, globosus, emboliformis, and fastigious. Okay, so DEGF from lateral to medial, dentatus, emboliformis, globosus, and fastigious. Okay, from lateral to medial, this intracerebellar nuclei embedded within the white matter of the cerebellum. And this is histological picture. Okay, cerebellar cortex shows three layers: outer molecular, middle Purkinje cell layer, and inner granular layer. See, this is outer molecular layer. The pinkish colored one is the outer molecular layer. Next, this single cell layer containing the Purkinje cell bodies. This is known as Purkinje cell layer. Okay, Purkinje cell layer, middle Purkinje cell layer, and inner granular layer. This violet dots, they are forming the inner granular layer. Okay, and this is white matter. So these three layers are together forming the cortex, and here is the medulla. Medulla is formed by the white matter. Okay, white matter. So we will discuss these individual layers in details. First is molecular layer. This is molecular layer, the pinkish colored one. It lies just deep to the pyometer. Outer covering is the pyometer covering, and it is lightly stained layer with hematoxylin and eosin stains. And the cells in these layers are stellate cells in the superficial part. This stellate cell in the superficial part, and few basket cells in the deeper part. This is basket cell in the deeper part. Okay, so these are the cells of this molecular layer. And fibers are numerous branching processes of the Purkinje cells. These are the numerous dendritic processes of the Purkinje cells forming the tree-like branching. Actions of the granule cells. Okay, this is granule cell. Actions are entering into this molecular layer, and they are bifurcating, T-like branching occurring, and they are forming parallel fibers in the molecular layer. Okay, so actions of the granule cells and climbing fibers. The yellow one is the climbing fiber. They are reaching up to this layer, molecular layer. Okay, so these are the cells and fibers of this molecular layer, and it is lightly stained layer with H stain. Next is Purkinje cell layer. This is single row of Purkinje cell bodies that are arranged between the molecular and granular layer. These are single row of Purkinje cell bodies forming the Purkinje cell layer. And Purkinje cells are large flash-shaped neurons. Okay, you can see the shape. It is flash-shaped neuron. Dendrites of the Purkinje cells arise from the apex of the flask and enter into the molecular layer. So these are the dendrites entering into the molecular layer. And single action arises from the base, and that passes through the granular layer and white matter to the cerebellar nuclei. Okay, single action. This is the action of the Purkinje cells. They are passing through the granular layer and white matter of the cerebellum and going towards the cerebellar nuclei. This is only efferent of the cerebellum. Next, we are going to the granular layer, the light yellow colored portion. This is granular layer. It lies between the Purkinje cell layer and the white matter. This bluish colored one, this is white matter. So it stains dark. This granular layer stains dark with hematoxylin because of closely packed numerous small granule cells. Okay, these are the granule cells, closely packed numerous granule cells. Mainly the nuclei of the granule cells appears like the dark staining dots. Okay, dark staining dots are the identification feature of this granular layer. Okay. Cells of this layer are granule cells and Golgi cells. These are the granule cells and here is the Golgi cell. Okay. And the granule cells, these are small neurons with rounded nucleus. Okay. And Golgi cells are large and stellate cells. Okay. Multiple processes are there. That's why stellate cells. So what are the fibers of this layer? Granule cells has mossy fibers that brings information from pons to the granule cells, pontocerebellar fibers. See, this granule cells has mossy fibers. It is anastomosing with the mossy fibers. The green colored one is the mossy fibers that brings information from the pons to the granule cells and also known as pontocerebellar fibers, these mossy fibers. And there are climbing fibers. Climbing fibers are this yellow one reaching up to the molecular layer. The climbing fibers and actions of the Purkinje cells pass through the granular layer. Okay, so these are the fibers of this layer. These are the different cells, stellate cells. All the cells we have got in these three layers we are discussing. So stellate cells lies just beneath the surface of the cerebellum in the molecular layer. These are small neuron and confined to the molecular layer synapse with the parallel fibers of the granule cells. Okay. And actions of the stellate cells confined to the molecular layer synapse with dendrites of the Purkinje cells. Okay. Next is basket cells. Basket cells are located in the deeper part of the molecular layer. These have small nucleus and they synapse with the Purkinje cell, these basket cells. And few dendrites synapse with the parallel fibers, bodies of the granule cells, Purkinje cells, clamping and mossy fibers. So these are the dendrites are synapsing with the basket cells. Next is Purkinje cells. Purkinje cells are you get in the Purkinje cell layer. It is large flash-shaped cells and they arborize in the molecular layer. Okay, and it has a single action to deep cerebellar nuclei, going towards deep cerebellar nuclei. Next, we are going to the granule cells. Granule cells are located in the granular layer and this has small spherical neurons and dendrites form the glomeruli with the mossy fibers and actions run parallel to each other and enter into the molecular layer. Golgi neurons lies in the superficial part of granular layer and these are large stellate cells converging inhibitory neurons and dendrites enter the molecular layer synapse with the parallel fiber. Action ends in the glomeruli. Okay, so these are the different cells and their axons and dendrites. So what is glomeruli? So this is glomeruli formation. Mossy fiber ends as dilated terminal. So this is mossy fiber end and this dilated terminal is surrounded by dendrites of the granule cells. These are the dendrites of the granule cells and actions of the Golgi cells. This is action of the Golgi cells. Okay, they are together forming the glomeruli. This mossy fiber terminal with surrounding dendrites and actions form the light stained area in the granular layer and these areas are called glomeruli of the cerebellum. Okay, so that's how glomeruli is formed. Next, we are going to white matter. White matter is composed of fibers and neuroglial cells. The deep pink portion in the middle, this is forming the white matter formed by the fibers and neuroglial cells. And the violet dot you are getting here, these are the nuclei of the neuroglial cells. Okay. So like this you will see under the microscope. So there will be outside diameter covering around the folium of the cerebellum. Then you will get the cortex. Okay, cortex. This outer pink portion. This is the molecular layer. Then this dots. These are the cell bodies of the Purkinje cells. So this is Purkinje cell layer. Then the violet dots you are seeing, violet dots, these are forming the granular layer. So these are the three layers of the cortex, and inside the cortex here you are getting the matter, getting the white matter. Okay. So, and this is enlarged view. This pink portion is forming the molecular layer. This is Purkinje cell layer, and this is granule cell layer. Okay, so these three layers of the cortex in enlarged view. So outer gray matter or cortex, it is formed by molecular layer, Purkinje cell layer, granular layer. Outermost layer cells are the stellate cells, basket cells, and fibers are the dendrites of the Purkinje cells, clamping fiber and parallel fibers. Purkinje cell layer is the middle layer, single row of flash-shaped Purkinje cells are present here, and granular layer is the deep layer. Cells are the granule cells, Golgi cells, and fibers are the mossy fibers and the glomeruli. Okay, so these are the three layers. And inner white matter is formed by the different fibers and the neuroglial cells. And what is glomerulus? It is enlarged terminal of the mossy fibers plus the dendrites of the granule cells and actions of the Golgi cells. Okay, so this chart you must remember for histology of cerebellum. It is important for your theory short note. Okay, also important spotter slide. Okay. What are the afferent fibers of the cerebellum? Clamping fibers. Clamping fibers are reaching up to the molecular layer. They are olivocerebellar fibers, actions arising from the inferior olivary nucleus. And they synapse in the molecular layer with the Purkinje cells, one fiber for one cell. And mossy fibers. Mossy fibers are other than olivocerebellar fibers, and they include the vestibulocerebellar, pontocerebellar, and spinocerebellar fibers. And mossy fibers have dilated terminal in the granular layer, and they synapse with dendrites of the granule cells, actions of the Golgi cells, and this synapse form light staining cerebellar glomeruli. Okay, we have already discussed the glomeruli. Okay, so outer cortex, so it is summarization for how to identify in the histology slide. Outer cortex or grey matter, inner white matter. Grey matter, three layers, outer molecular, middle Purkinje cell, inner granular layer. Molecular layer is lighter on He staining. It has numerous nerve fibers, stellate cells, basket cells, neuroglia. Purkinje cell layer consists of single row of flash-shaped Purkinje cells here, and granular layer is dark on He staining, numerous small granule cells and large Golgi cells. White matter is composed of fibers and neuroglial cells. So you have to write down these identification points in your histocopy. Okay, identification point for cerebellum. Cerebellar folia with outer cortex and inner medulla or inner white matter. And cortex has three layer structure: outer molecular, middle Purkinje cell layer, inner granular cell layers in. And main cell types are Purkinje cells and granule cells, more numerous. These two cells are more numerous. And medulla is made up of the white matter. Okay, so this is identification points of the cerebellum. Diagram you draw for cerebellum in your histocopy. Okay, outer diameter covering here. Then this is the molecular layer, Purkinje cell layer, granular layer, and this is inner medulla formed by the white matter. Okay, so this diagram you draw in your histocopy. You have already drawn. Okay, so you draw this diagram in your histocopy. Okay, for cerebellum. Okay, so prepare histocopy and prepare as a short note. Okay, so in our slides in our histo lab, we have the slides of cerebellum. You go and see. You can properly differentiate the molecular layer, pink molecular layer, and the violet dot collection, those granular layer. You can differentiate, and this Purkinje cell layer are not that much prominent. Okay, you go, and white matter is very thick. Here you have seen very thin amount of white matter, but in our slides, white matter area is very thick. Okay, so you go and see. I am coming. I will explain. Okay. Violet covering, outer violet covering is the pyometer covering. Okay. After that, you will getting a pink area. That pink area is the molecular layer. Okay, molecular layer of the cortex. Purkinje cells are not much prominent. After this pink area, you are getting multiple a layer which is formed by multiple violet dots. That is the granular layer. Okay. After that, you are getting a light pink area. Whole of the area is forming the medulla or inner white matter. Okay. So you see this slide. This is those layers of the cortex you can see prominently. Some of the Purkinje cells is visible. Large nuclei. Okay, at 7 o'clock position, one large cell you can see at the junction between the molecular and granular layer. That is the Purkinje cell. One Purkinje cell is visible here. Okay, so you see this. Folium is located like this. Outer pink violet covering is the pyometer. Here also you can see folium is lying like this. Outer violet covering is the pyometer covering. Don't slide the field. Okay, see in this location only. Okay, so outer pink violet portion is the pyometer covering. After that, you getting thin pink area. Okay, that is molecular layer. Then some dots, large nuclei. Large nuclei are of the Purkinje cells. Okay, large nuclei are the Purkinje cells. Then some violet dots, areas of violet dots, consolidated violet dots. This is granule cell layer. After that, the whole of the pink area on this side, you are getting that is the white matter. Okay. Here also you see this. Here also same thing. Folium is lying like this. Okay. Okay, come here everyone. Come here. Okay. Pen আছে কিছু? কিছু নেই না pen কিছু? Okay. So see here. This slide is looking like this. So this is the folium. Outer covering is the pyometer. Okay. After this pink area. This pink area is the molecular layer. Okay. After that, এখানে দেখ কিছু large violet dots are there. Here one, এখানে একটা one, here one. These large dots, these are the nuclei of the Purkinje cells. Then consolidated violet area, this violet dots, these are the molecular layer. Sorry, this is the granular layer. Granular layer, outer molecular, then Purkinje, then granular. Granular layer is consist of violet dots only. And whole of this area is forming the medulla or inner white matter. Okay, so this is folium covered by the pyometer. This violet covering is the pyometer. This is pyometer covering. Here, this is the pyometer covering. এই যে এই বাইরে এইখান থেকে এই হোয়াইট এরিয়াটা যতদূর ওটা পুরো pyometer covering. ওকে, তারপরে যে পিঙ্ক এরিয়া ওটা হচ্ছে মলিকুলার লেয়ার. তারপরে এই যে কিছু ভায়োলেট বড় বড় ডট দেখতে পাচ্ছিস, এই যে এখানে 1 2 3 4, these are the Purkinje cell layer. তারপরে এই ভায়োলেট ডটস এটা গ্র্যানুলার লেয়ার, এন্ড তারপরে হোল এরিয়াটা মেডালা অর ইনার হোয়াইট ম্যাটার. এটা মেডালা. মেডালা আর ইনার. এটা কি ম্যাম? কোনটা? এটা another folia or white matterary folding হবে. এখানে actually multiple sections are there and they are overlapping. That's why single folia আর কমপ্লিট ফোলিয়া ক্যানট বি ভিজিবল, এটা পার্ট অফ দ্য ফোলিয়া, এই দিকটাতে কতটা বেশি ভিজিবল নয়। ওকে,
যেটা স্টেনিং কম থাকবে ওটা হোয়াইট ম্যাটার ধরে।
হ্যাঁ যেটা স্টেনিং কম থাকবে, যেটা লাইট পিঙ্ক ওটা হোয়াইট ম্যাটার। ওকে ওকে, এটা দিয়ে দে। ওকে